Okay, hi semua. Now we will go to the last part for the topic of genetic inheritance, and this will be about the genetic mapping. So what is genetic mapping? It is actually a method to determine the position or the sequence of genes along a chromosome. For example, if there are two genes on this one same chromosome, let's say gene A and gene B, we will do this genetic mapping procedure in order to determine the location or the distance between these two genes. And the distance between the linked genes will be reflected by the crossover value or the COV. There is a formula to do this genetic mapping. The formula will be the crossover value will be the number of recombinants divided with the total number of offspring and we time with 100. In order to calculate, we need to have the data from a test cross involving linked genes with crossover. And if 1% of the value of COV, this shows that the distance between two linked genes is 1 map unit or 1 centimorgan. For example, if a test cross was done on a Drosophila species with grey body and normal wings, this will produce 4 types of offspring. This may be the data given from this test cross. You'll be able to see that there are 4 types of offspring and there will be 2 types with a large number. This will be the group that will be having the parental phenotypes. Meanwhile, another 2 groups will be in a lower number. This will be the recombinants. And so we will start by calculating the crossover value, in which we get the number of the recombinants over here, 206 plus 185, and that equals to 391. And we divide this 391 with the total number of offspring, which is 2300. And then we times with 100. And the crossover value will be 17%. And so the distance between the genes will be 17 map units, or you can also say that the distance will be 17 centimorgans. Based on the recombination data, the position or order of the genes along a chromosome can also be identified. For example, let's say there are these genes P, Q, R, and S, and all the genes are linked on one same chromosome. We might get the value of COV. For example, P and Q will be giving the COV value of 35%, R and Q will be giving the value of COV of 40%, R and S will be giving the value of COV of 30%, P and R will be giving the value of COV of 5%, while Q and S will be giving the value of COV of 10%. And so we start with the highest percentage, which will be between R and Q. And so the sequence for the genes would be R and Q with the longest distance of 40 map units. And then we will continue with the second one, which is 35% between P and Q. Then we continue with R and S having the COV value of 30%, R and S. And then we continue with the Q and S with the COV of 10%, and finally the shortest distance between P and R of 5% only over here. And so we can conclude that the sequence for the genes would be R, P, S, and Q. And so you can try to do some other exercises and questions concerning this genetic mapping. See you guys later. Bye bye.